Okay, so good morning, everyone, and welcome to today's webinar hosted by Gator Bio in collaboration with NovaRock Biotherapeutics. The webinar is titled Evaluation of Gator Pro, a high-throughput BLI system for kinetic analysis. My name is Isha Trehan, and I'm a product marketing manager at Gator Bio, and I would be moderating today's webinar. Before we get started, I would like to go over a few housekeeping items so you all know how to participate in today's event. All attendees are listening in using the computer speaker system by default, and all the attendees are currently on mute, and the in-meeting option for chat is currently disabled. However, you can message the moderator at any time in case you're having any technical difficulties. One will also have the opportunity to submit text questions to today's presenters by typing in your questions in the Q&A box that you would see on your screens. So you may send in your questions anytime during the presentation. We will collect these questions and address them during the Q&A session, which would be hosted end of today's presentation. Quickly getting into the format of our presentation today, of the webinar today, we have both our speakers present here who would be presenting for the next 45 minutes. And once they finish the last 15 minutes, there would be a quick poll on your screens that we would kindly ask you to participate in. And then we would follow up with all of our Q&A sessions for the next 15 minutes. So let's begin. And I would now like to introduce you to our speakers for today. The first speaker for our webinar today is going to be Rich Wang. He is the product manager at Gator Bio, and Dr. Huarui Lu, principal scientist at NovaRock Biotherapeutics. I'm handing it over to Rich Wang and our first speaker of today to start the webinar. Thank you.
Thank you, Isha, and good morning, everyone, and welcome to today's webinar. And thank you for your joining. I'm going to introduce the Gator Bio about who we are, what we do, and what technology we are using, and also what exciting product we are offering currently. Yeah. So Gator Bio is the leading BLI innovation company for the last 5 years. We focus on different applications such as quantitation, kinetics, epitope binding, small molecule analysis, and the gene therapy. We are the company headquartered in the Palo Alto, California, with the best team of the best mind of the BLI, including our founder and CEO, Dr. Hong Tan, our CTO, Robert Zug, and the director of R&D, Pu Li. With the best mind of the BLI, actually Gator Bio has a long legacy with the BLI lead technology. So back to 2001, Dr. Hong Tan founded the first BLI lead technology. And over the past 2 decades, actually BLI is getting more and more powerful, and it's a very powerful technology to expand the biotherapeutic discovery for the scientists. However, it has its limitation. And that's the reason Dr. Hong Tan and Robert Zug, they came back and founded this BLI technology, the next-gen BLI, Gator Bio. So as of today, Gator Bio is offering 5 different instruments: the Gator Pivot, Gator Pilot, Gator Prime, Gator Plus, and the higher throughput Gator Pivot and the Gator Pro. So what is BLI-less technology? So actually BLI-less technology is a technology based on the measuring in reflected light. So the partial white light beams are shining through these glass-made biosensors and hit each face of the boundary and reflect it back. So the change of these interference patterns allows us to monitor the real binding event of your target molecules. With our proprietary surface chemistry, the target biomolecule can bind to the surface directly. And so this real-time change of this interference pattern, in most of the case, this signal is proportional to the number and the size of the biomolecules. And our Gator instrument will pick up this biosensor, move from well to well on the microplates, where a scientist or a user, you can pre-fill or pipette all your sample into this microplate format. So actually, all these idea about this BLI idea can be explained in these 1-minute short videos. So as you can see, a white light means a light source shining through this glass-made probe, and these lights will hit each face of the boundary, reflected back, collected by our instrument. If one molecule binds to the surface, so we can observe this change of this real-time interference pattern got shift. So we can see and monitor the binding event. And of course, if another molecule binds to the surface, we can observe another nanometer shift and this interference pattern shift. So we can observe another binding event of your target molecules. Vice versa, if these binding molecules falling off from the surface, we can observe this interference pattern shift back. So we can interpret into this falling off and the downdrift signal. We call it as the dissociation phase. Pretty simple, right? So what is the Gator can do? Actually, Gator is a tool to expand your biotherapeutic discovery, ranging from early discovery down to manufacturing and QC, including antibody titer, yes-no binding, off-rate ranking, epitope binding kinetics, and quantitation. So Gator Bio, we are offering this total solution as a whole package. This includes 3 parts. The first one is our Gator family instruments, the Gator Bio biosensors and the consumables, and of course our Gator One software. So let's jump into the very first part of this total solution. The first one is this biosensor, our consumable. We are offering the full suite of the biosensor to meet all the users' applications needs. And we can divide into 2 different areas, the antibody characterization and the AAV analytics. In the very first part of this, our biosensor applications, we include such of those label-free biosensor, protein AGL, human FC biosensor, mouse FC biosensor, or arming reactive biosensors. And we also offer some of the popular and common use purification tagged biosensor, such as anti-His, nickel NTA, and we are offering this anti-PEG biosensor for the quantitation and the kinetics of the pegylated lipid-based biomolecules. And we also offer the full suite of the assay-based biosensor to meet different applications needs. We are also offering the only solution from the upstream to downstream for AAV analytics, including the titer determination and the ratio measurements. We never stop serving our customers. So actually we are offering a different type of the custom biosensors, including the anti-flag, anti-strep tag 2, anti-rabbit, anti-rat, and RBD biosensor. And these biosensors, actually, you can, as a user, it's very easy to log into our portal and to place this order. And we also provide the technical and scientific support if you have the more advanced or a different type of the custom biosensors you would like to bind to. So here, the second part of our Gator total solution includes this Gator family portfolio. So right now we are offering 5 different instruments on the market, from the lower throughput up to the higher throughput. Starting from the entry-level Gator Pilot, this 4-channel system, which still is a very friendly tool, and also can provide a very reliable 4 channels simultaneously read. So as a scientist, you can still get a reliable kinetics, full kinetic data. And we also provide this Gator Prime, plus they are all 8-channel systems. With a higher throughput, we are offering this Gator Pivot with a 16 channels simultaneously read and equipped with a 2 flexible microplates format. It can either be a 96 or 384 well plate. And this Gator Pivot is also equipped with evaporation control and the sample cooling features. If you are looking for a higher throughput system, Gator Pro is your answer. It equips up to 32 spectrometers with 32 simultaneously read with 3 flexible plate format. Either it can be the 96 or 384 well plate, which allows the user can run up to 150 samples per batch. Let's dive into a little bit about how we make this higher throughput. So as this slide you can see, let's take this Gator Pivot as an example. This Gator Pivot actually is equipped with 3 plates, but 2 of them is designed for the sample microplate. It can either be 96 or 384 well microplate format. So as a user, you can pipette all your sample into this 96 or 384 well plate, and our instrument will take care of the rest of the measurement. It's all automatic. The last part of our total solution is this Gator One software, which supports different applications such as quantitation, kinetics, epitope binding, and the viral factor analytics. This software is very, very easy to use, and it's also the real-time monitoring the change of this interference pattern. So allows the user to see the real time of your binding event of your target molecules. So after this total solution, so what Gator can offer and Gator's advantage. So Gator actually, we are offering a higher throughput and much faster workflow for the user. And then the ease of use of this Gator solution so allows a user to run either the purified or crude sample. And the best part is since it's a non-fluidic system, so it's a very, very low maintenance. You don't need to worry about any clogging or any maintenance changing the tube issue. With our robust design of this biosensor and with our proprietary surface chemistry, those biosensors can be reused several times to provide a higher value of each run, and not to mention this wide range of our innovative applications. Yeah, so thank you for your listening, and I'm turning to Dr. Huarui to introduce his work.
Okay. Thanks, Rich, for the introduction. And it's my pleasure to have this opportunity to share some Gator Pro data. So as Rich mentioned, Gator Pro is a new high throughput BLI instrument that Gator Bio launched recently. And Rich just gave us a very nice introduction and about this kind of technology and machine. Next. So before I start in the data sharing, I would like to give a very brief introduction of our company, NovaRock Biotherapeutics. NovaRock is a clinical stage biotechnology company that founded in 2018. Our company focuses on the development of innovative antibody therapy for cancer and autoimmune disease. We already built up some different platform for antibody discovery, characterization, engineering, and optimization, and cell line development. So you may check our website for more information if you're interested. So briefly, in our pipeline, we already had some molecules advanced into the phase 1 clinical trial stage. and some got FDA approved of IND application, and some are still in the preclinical stage. They covered different fields such as oncology, immuno-oncology, and immunology field, and a variety of targets for different indications. We collaborate with different company, Leap Therapeutics and CSPC for different projects. So in our company, other than the classical, ELISA cell-based binding assay, we also have BLI SPR platform for in vitro, antibody characterization. Currently, we had Octet, Gator Prime, and the old version SPR machine vehicle 3000, functioning and operating for some specific, aims such as binding kinetics, binding affinity, epitope binding, and sometimes, quantitations. The main application, for the BLI in our companies that are in the kinetic analysis and epitope binding and cross-blocking, and sometimes I mentioned, quantitation for the protein titer, protein induction yield, something like that. As Rich mentioned, the BLI is one of analytical tools used routinely in our facility to for kinetic analysis in antibody discovery and characterization. With an update, Gator Pro launched the new high-throughput BLI system, Gator Pro, recently. We cooperated with Gator Pro to evaluate this new machine for the common application like kinetic analysis. We knew the BLI system have some advantage, such as like label-free, real-time response monitoring, and reagent saving, and possible, recovery. And a schematic, diagram on the left side just showed the general process for the binding kinetics. Basically, after the baseline, antibody was captured onto the appropriate probes, then going to analyze for association and dissociation. So we run a bunch of different applications on the new Gator Pro instrument with high throughput format. That means 384 well plate format. Even we had not that much of samples at that moment, but that easy to scale up if you have more sample. Basically just have some the plate designer and the pipetting works. We run limited number of samples with Gator Pro and compared with the Octet. Back one slide, Rich. Yeah, side by side, then we run binding kinetic use the Gator Pro only. We also run other different, applications such as epitope binding and cross-blocking, and check the protein titer yield. So I will focus on the first two parts. I'm not going to talk too much about the other applications. So next. So now I'm going to share with you the first experiment that's binding kinetic, performed on the Gator Pro and Octet side by side with a limit. I mentioned, the numbers of sample. Basically, the the principle of experimenting performed as described here. We use antibody capture format, use HFC probe, capture antibody, and then test different concentration of analytes. That's the protein antigen, and we analyze data use instrument built-in for Gator that have Gator 1, software, Rich mentioned. For Octet, they have for the bio data analysis software. We use a 1-to-1 model. Next. So first of all, we test total 14 antibody by Gator Pro and Octet side by side. So the binding parameters are summarized as the left side table. We ranked those antibody based on we rank the KD value of antibody based on the Gator data. We can find in general, the KD value get from Gator Pro a little higher than an Octet, but the change less than like 20%. We found, the KD value was extracted from both Gator and Octet are in the same range and the similar trait as the color, highlighted. That's ranking. So we also graphed the KD value from Gator Pro and Octet, and found all the dots are distributed orthogonally. That's indicating they are highly, correlated. Same thing we can see if we graph the off rate and on rate for all 14 tested antibody. Both are the dot distributed orthogonally too. That's correlated. Next. So other than the summary table showed before, we also, the following slides are showing all the fitting curve. Gator, on the left side and Octet on the right side. We can clearly see the good response for both, instrument for every, tested antibody, and the good fitting curve for both instrument. Next. Okay, this other, antibody. We can clearly see the same thing, same phenomena. Yes, same. So from above, experiment, we found that the high throughput format run by Gator Pro. Again, I need to mention, we just run limited numbers of samples at that moment. So for our testing, antibody showed good fitting response, a good response and good fitting, and correlate with the Octet data. So after that, next we just run some kinetic analysis by Gator Pro only to see how it works. Basically, this experiment runs the same format. We still use the antibody capture format. And this time, total 16 samples were tested on the Gator Pro for the binding kinetics. As the summary table showing here, so all the tested antibodies show good response and a fitting curve with R, square that's close to, very close to the 1. That's the parameter usually instrument, for quality, check. Yeah, this slide just show, other than a summary table, we show the fitting curve again, tested antibody, show good response and good fitting with the different on-off pattern. So next. Okay, just brief summary. We can overall, in our hand, we test limited numbers of samples, but those all tested antibody, at least in our hand, shows good binding fit. Use the Gator Pro, kinetic analysis. Also, compared to the Octet, that's common instrument in the market, the kinetic parameter from Gator Pro are in the same range and similar in trade, also are highly correlated with Octet, measurements. Next. Yeah, basically that's my, the short, presentation for the some application, we performed on the Gator Pro. That's new instrument, launched by the Gator Bio. I will thank to our colleague Zhiqiang Zhu and Dishen, a doctor, and also thanks to Gator Bio Rich and other to organize and give this opportunity to present some the interesting data. Thank you. If you have any question, we can take some questions.
Thank you, Dr. Huarui, for a great presentation. We are now launching our polling questions, which would be on your screens. Please select the appropriate response. And, once we'll probably give it about 2 minutes for you to answer the questions, and we can move into the Q&A section. The first question is for you, Dr. Huarui, and the question is, what was your antibody loading level in the Gator versus Octet study?
Okay, that's a good question. Usually, we loading level that's between 0.6 and up to 1 nanometer. That's good, the surface density for the kinetic analysis.
Perfect. The next follow-up question was, and was the antibody loading levels similar between the Gator and Octet?
Yes.
Okay. The next question is, which probes were used on the Octet instrument for this study?
Oh, that's also, human anti-human FC, probe.
Perfect. The next question is Gator 1 an add-on cost, or does it come with the device purchase?
That's a very good question. It comes with the device purchase. So you don't need to pay additional cost for this Gator 1 when you first purchase the Gator system. Yeah, and of course, I believe our sales team, they are very happy to answer the question. But in general, you don't need to pay additional cost, because the instrument should always come with this analysis software for our scientists to do their data and their works.
Okay, thank you. There is a follow-up to the same question. So does the Gator 1 software perform the epitope binning analysis? And if so, would you be able to provide an example of the output so we can evaluate the feasibility of importing these results into LIMS?
Yeah, this is a very good question. Yes, Gator 1 software, of course, it supports this epitope binning analysis, and it supports this including the cluster and the matrix, and also some of these bin chart. And to support this LIMS, I believe we are very interesting to see which type of the platform you are using, and also if you would like, we are very happy to have another conversation, and then we can show you what is the output looks like, and if this is something that you're looking for to import to your this LIMS systems.
Thank you. there is the next question, which is, did you compare results from Octet and Gator with SPR just to see which one is more closer? This one's for you, Dr. Huarui.
Oh, for this, specific, experiment, we didn't, compare to SPR, but we do, compare SPR and BLI, different technology before, that's quite, other than, the experimental variation, that's quite, the very, highly correlated in the same range, similar, trait. We are not saying that's exactly same, for the rankings that have some variation.
Okay. This one should be for you, Rich. So is our Gator One software continuously being upgraded and improved? If so, are the upgrades also at a cost?
Yeah, this is a very interesting question about this one. So from what I can tell, yes, Gator One is always upgrading all the time because we want to support our customer to do all their applications. And like the presentation I presented earlier today, actually, we never stop this innovation. So of course, to support this innovation, we have to keep upgrading and keep updating our software and to support our customer. And in terms of the cost-wise, I would say as of today, for all these purchase of the Gator instrument, then currently it's kind of free of charge of this upgrade. Yeah.
Okay. And then I think there's a follow-up to the software question. So can the Gator One software be used on multiple machines, or are licenses required?
Yes, it's a license required. Yeah, but to answer this question, so I can share some of the idea because we understand nowadays a lot of users, they are working from home, they do their studies in a lab, and then they went back to their cubicle, or they would like to analyze their data from anywhere. So actually, the one purchase of the Gator instrument, it always comes with a 3 free license. So which means that as a user, you can install you can have this instrument like a workhorse running your experiment all the time. But meanwhile, you have additional software license to be installed on other PC, so that you can analyze your data anywhere, anytime.
Perfect. I hope that answers the question. There is another question, which is from the graphs, and this is for you, Dr. Huarui. So from the graphs, the association times look different between the Gator and the Octet. Was this the case, and why? Oh, 1 second, we can't hear you.
Dr. Huarui, you are muted. Yeah.
Actually, that's the same association dissociation, but just because when we extract the graph that have a different profile, we modify the graph, that's the same.
Okay. The next question again is for you. Is there a comparison of Gator to BLI in the PM range?
Unfortunately, that's what we have. For this experiment, that's not down to the PM range.
Okay. This one could be for either of you. So there is a question regarding our biosensors. So do you have other biosensors such as for glycan analysis, HCP analysis, etc.?
I think I can answer the question. Yeah. So currently we don't have these two type of the biosensors at this point, but I believe this is something that we will keep our eyes on that and always be innovative and always innovate these different biosensors to meet our customers' need.
Thank you. Okay, so the other question again is for you, I believe, Rich. So do you have any suggestions of the nanometer shift signal for evaluating kinetic assay?
Yeah, this is a very good question. Yeah. I would say in terms of this nanometer shift signal, then I will divide it into two different parts. First one is the loading capacity, like the question we got before, and Dr. Huarui mentioned that the loading nanometer shift signal, we will always recommend it as long as it's above like 0.6 or up to 1 nanometer shift is enough. And the rule of thumb of this loading nanometer shift is always that as long as it can give you enough signal for your following association state, then it's a good loading signal. So usually we don't want to overload the biosensor because we don't want to have any ability or any steric hinges on this ligand to be immobilized on the surface. And the other one is the nanometer shift number. So when we are talking about this association phase, then of course our system's background noise of the background is around 0.3. This 0.3 nanometer. So as long as you can get this signal to noise ratio is big, then of course you know that you can determine it's a high signal, it's a good signal to allow you to have this reliable data.
Thank you. I think we just have a couple more questions. So this one again for you, Rich. Can our biosensors be recycled?
Recycled, yes. This is also a good question. I would say because those biosensors, we use another term, more like a reusable or regenerable. So regenerable, it means that actually those biosensor is not a one-time use, and after you use that, actually you can just regenerate the surface, and our surface chemistry will stay on the surface. So you can reuse the biosensor over and over again. And unfortunately, because these most likely the biosensor and those samples are biohazard. So after you use them, I believe you should still throw them into this biohazard to do the trash recycle or anything. Yeah. However, I believe we are also trying to do another way is, for example, we do have this reusable, we call it as a reactivable strap evident biosensor, the flex assay. So with the multiple times of the reuse of these biosensors, actually it's also the other way to turn things into green. Yeah.
Perfect. Thank you. Another question from someone who says, can you please explain automation compatibility of Gator Pro?
Yeah. Explain this. So right now, of course, we are offering this automation, this API portal with the vendor. And so, of course, this automation is available for both Gator Pro and Gator Pivot. So as long as if you are looking for this automation and to be integrated, we are more than happy to help and also have this conversation with these automation vendors.
Perfect. The next question is, what is your suggested max runtime for an assay if one doesn't have evaporation control?
Yes, this is also a good question. So in our experience for those max runtime, it depends on which microplate format you are using, and of course the running temperature you are using. For example, if you are using a 96 well microplate, of course the area of the well is bigger than the 384 well plate. So we would recommend not to over 6 hours, 7 hours this runtime. And of course, on the other hand, the 384 well plate, because the microplate area is much smaller, so the evaporation effect it's it got reduced a little bit. So of course, the runtime we will always do we can recommend around 8 hours this type of runtime. But of course, most of these runtime, how do you make sure it's data, whether the data or the runtime is good or not, still depends on which application you're looking for. For example, if it's a screening, you are looking for a very, very high throughput, then the data, the same trend, it will not affected by this evaporation. So you can expand this runtime. However, if you are doing and you are looking for a very, very reliable and very robust data, so we will still recommend to do a very shorter time to run this whole experiment, because you don't really want to have another parameters to affect your data.
Thank you. I do see 2 more questions pop up. So someone's asked, what is the cost of the Gator Pro?
That's a very good question. Yeah. So I think this one we can discuss this after this call, and we would feel free to contact us, and we will always love to provide this information.
Perfect. And I think the same question is, what is the cost for the Gator pivot?
Yeah, I would say this will be the same answer. Yeah, we are very, very happy to answer this question after this call. Yeah, so feel free to leave your information, or feel free to contact us. Yeah.
Perfect. We did not have any open questions at this time. If anyone has any questions, can you please post it in the Q&A box? We would be happy to answer them for you. Okay, there is actually another one. So maybe this one's for you, Rich. What is the good quality of data or the signal from the analyte?
Yeah. Like I previously mentioned, a good signal of this from the analyte, which means this so-called association. And if we are talking about this kinetics data analysis, our system's background noise is a 0.03, this range. So as long as you have a high enough signal to noise ratio, then you can trust the data. For example, let's say, and this most of the case, this signal is proportional or very, very highly correlated to the size of the biomolecules. For example, if you are having a bigger size of the molecule, like the antibody, then of course we are looking for around at least the highest concentration of your analyte should be around at least 0.5 nanometer, this kind of range. However, if you are looking at the small molecular analysis, and then you already know they are very, very small target. So maybe you can see it's as long as the highest concentration can reach like a 0.1 nanometer, then it's also, we also consider as a good signal, because it really depends on what is the size of your molecules and the signal to noise ratio to determine whether it's a reliable data or it's just a non-specific binding or it's just purely the background.
Perfect. Thank you. If there are no questions, we can go ahead and conclude today's webinar. Let's wait for a couple moments. Okay, so we do have a question. Can you explain the benefit of your recently launched 96 well tilt plate? And will you have a 384 well version as well?
Yeah. So the 96 tilt, the benefit of this 96 tilt is that because, for example, one of the benefit of this 96 tilt plate is you can reduce your sample requirement. So the normal 96 flat microplate, either from we recommend our user to use either grinder or you can use our Gator 96 flat microplate. But with this tilt microplate, actually you can reduce the volume from 180 microliter per well with the 96 flat microplate down to 130 microliter per well for this 96 tilt microplate. Yeah, and in terms of this 384 well format, yeah, we would like to keep in touch with you, and please stay tuned on our website or our linking.
Perfect. We do actually have a participant who would want to ask you a question.
Yeah.
Sean Moraine, I do see you have your hands up. I will allow you to talk and okay, I think you should have access now, Sean. I'm not sure.
Sorry, I don't have a question. I hit that button by accident.
No worries. Thank you. Okay, so if we do not have any more questions, I would personally like to thank our speakers for today, Rich Wang and Dr. Huarui, for taking out the time and actually sharing their research and a lot of information about Gator Bio. And I thank you everyone for attending today's webinar. If you do have any other questions, please feel free to contact us at info@gatorbio.com. And if you have any questions regarding any of our instruments or cost and pricing, please feel free to reach out to our sales teams at sales@gatorbio.com. And once you leave today's webinar, you all would be receiving a follow-up email within the next 24 to 48 hours with a link to view a recording of our today's webinar. Please keep an eye for our upcoming product launches, updates, any information regarding what's happening at Gator Bio and future webinars. We are very actively engaged on LinkedIn as well as other platforms. Follow, like, and subscribe, I would say. And on behalf of Gator Bio and NovaRock Therapeutics and our presenters today, thank you for joining us today and have a good rest of your day. I would give you 8 minutes back in your day. So have a wonderful day today. Thank you. Bye-bye.
Thank you.
Yeah.
Okay, thank you. Bye.
Thank you. Bye-bye.